So upon integrating the equation, we'll get finally the partial molar volume into difference pressure P2 minus P1, which will be equal to minus RT ln AW, or activity of water, where VW is assumed constant. Then the pressure difference P2 minus P1 by definition is known as the osmotic pressure. So this is osmotic pressure represented by capital pi. So VW pi or will be equal to minus RT ln AW, or pi will be equal to RT by VW ln AW. And RT by VW, PV equal to nRT, you know the same relationship we got here. For calculating the activity of water in the solution AW, the vapor pressure data are needed. Upon AW is calculated from the relation. So vapor pressure of water in equilibrium and Pw star, that is at same temperature, it is the vapor pressure of pure water. This is the vapor pressure of water in equilibrium with the solution at a specified temperature, while Pw star is the vapor pressure of pure water at the same temperature. So activity will be the ratio between the vapor pressure. If the solute is volatile, the partial vapor pressure water must be used for Pw. So substituting all these things, we will get pi into Vw, that will RT ln Pw by Pw star. Or so anyway, finally what we will get, pi V equal to CRT, or pi equal to CRT, where C is the molarity of the solution. So that is known as the Weintraub's equation for ideal solution. This is also we have known in our higher secondary. For electrolytes which ionize in solvent or water, the observed osmotic pressure is more than that of the predicted from molar concentration data as the osmotic pressure. So it is a colligative property. Then we have to apply the Van 't Hoff factor. Okay, so Van 't Hoff factor I, that is equal to the observed colligative property divided by the theoretical, experimental to theoretical, that is the Van 't Hoff factor. So Van 't Hoff factor is approximately equal to the number of ions produced during ionization per molecule of the electrolyte. So this is the relationship. Then we'll move the reverse osmosis process. So let us have a little 5-minute break, then we'll come. Okay, we'll start. So, reverse osmosis. Osmosis is a natural process wherein solvent or water passes through the semi-permeable membrane from the side with lower solute concentration to the higher solute concentration until the equilibrium is established. But when the flow of water is reversed, as a pressure difference which is greater than the osmotic pressure is applied in the solution side, then as a result of this, separation of water from salt solution becomes possible. This phenomenon is termed as reverse osmosis. It is also known as hyperfiltration. The process is generally evaluated in terms of parameters such as water flux (Zw), solute separation (R), and water recovery (Y). The definition of these parameters are given below. So the parameter water flux (Zw) refers to the amount of permeating solvent or water per unit time per unit area of the membrane. So by definition, Jw equal to Q P upon A and T. A is the membrane area, T is the time by which water quantity is the quantity of water permitting the solvent. Then the separation of salt by membrane is commonly expressed as solute separation R, which is the fraction of salt in the feed that does not permeate through the membrane. So it is 1 minus Cp by Cf. Cp and Cf denote the solute concentration in the membrane permeated, permeated stream and in the feed inlet stream respectively. And water recovery Y, commonly used to define the percentage of feed water that is converted into pure water in the permeate and is calculated as Qp by Qf into 100, where Qp and Qf denote the quantity of permeate stream and feed inlet per unit time. Then same basic equations we have to as we have derived for osmosis. It is the reverse osmosis. You have to apply more pressure than partial pressure. So P is the applied pressure. So like this. So this is the general equation. Jw equal to dw into Cw into Vw, partial molar volume, into P minus pi upon RT into del X. So the pressure difference P del X is the thickness of the membrane, dW and Cw refers to the diffusion coefficient and concentration of water in the membrane phase expressed as a volume ratio and which is dimensional quantity. Term V W bar RT refers to the partial molar volume of water, gas constant and temperature respectively. So like that if you calculate, so finally we will get this Js equal to A into P minus pi. So these are the equations which are used in reverse osmosis. Then the concentration polarization results from built of highly concentrated solute on the membrane surface as compared to the bulk feed solution away from the membrane surface. This occurs because water permeation at the membrane surface leaves the more concentrated solute layer which must diffuse back into the bulk liquid. So concentration polarization increases the osmotic pressure at the membrane surface which causes a reduction in water flux and an increase in the salt diffusion across the membrane. So at such high concentrations, colloidal materials in the feed solution may also agglomerate and foul up and foul the membrane surface. So the activity will be lost. The dialysis is a membrane separation process wherein solute they diffuse across a membrane barrier by means of a concentration gradient. The rate of diffusion of a solute through the membrane increases linearly with increase in concentration gradient and the proportionality constant, namely the diffusion coefficient. So D is a characteristic property of the solute for a given membrane. So dialysis is a highly constrained process because the molecular diffusional rates are so. Diffusional rates are slow, and if the receiving solution, that is the diazolate, are not continuously removed, the solute concentration on both sides of the membrane will tend to equalize, which will neglecting the driving force for separation. So these are the mathematical relationship. Then move to the electrodialysis. So electrolysis or electrodialysis is a membrane process wherein preferential transport of cations and anions are achieved during the cation and anion selective membrane as a result of electrical driving force. So using this process, the concentration of ionic species can be increased or decreased so that practical concentration or depletion of an electrolyte solution is possible. As shown in the figure, the counter-ion transport, co-ion transport, diffusion, and osmosis, these are the things, the various transport processes occurring in electrodialysis process. The counter-ion, that ion having opposite charge compared to that of fixed group of the membrane, the counter-ion transport constitute the major electrical ion movement in the process. Anion is the counter-ion for anion selective membrane, and cations are the counter-ions for cation selective membranes. The counter-ion formation through the membrane carries with them a certain quantity of water by electroosmosis. The co-ion transport, co-ion means ions having similar charges as that of fixed group of the membranes. So the co-ion transport is comparatively small and is dependent upon the quality of ion selective membrane and brine concentration. Anion is the co-ion for cation selective membrane and cation is the co-ion for anion selective membrane. Water is also transported electroosmotically with the co-ions. In diffusion electrolytes, diffusion of electrolytes occurs from the brine to the diazolate because in the electrodialysis process the brine is usually more concentrated than dialysate. Water transport is also associated with electrolyte diffusion. So it's the various transport processes already I have discussed. Then if you see the column efficiency, the efficiency of electrodialytic process is given in terms of Coulomb Coulomb efficiency eta, that is the number of equivalents of electrolytes displaced from diazolate to the brine divided by number of faradays of electricity passed in the process. So if you know the value of eta, then another is the rate of counter ion transfer flux, that is J, through the membrane of electrodialysis process is given by this relation, where I is the current density, ampere per square centimeter area of the membrane, F is the Faraday constant, 96,500. Similarly, the electrical ion flux J in the solution is given by this relation, this T prime T bar, this is rate of counter ion transport, this is the electrical ion fluxes. So Ti divided by F. So the other relationships you have to just check. Then the applications. An application of membrane separation process is used in, already we have discussed or we have some idea where it will be applied. Desalination of water treatment, protein recovery, production of table salt or from saline water, then hemodialysis, ion selective membrane electrodes, specific gas probes. These are the membrane and this ion electrode, typical membrane and commercial ion selective membrane electrodes. Specific gas probes, detection and analysis of particulate contamination, microbiological analysis. So these are the applications. Then questions. Then we'll move to the next chapter, the last chapter, that is the electrophoresis. Please give me time of 2 minutes. We'll come back. I will see it again. Okay, so electrophoresis. So that means this is also a membrane separation technique, refers to osmosis, or here the ions, respective ions, they will flow towards the respective electrode by application of electric field, and they will pass through the semi permeable membrane and can be separated. So that is electrophoresis. We'll discuss something about electroosmotic flow, then basic principles and operation, different forms of electrophoresis, slab electrophoresis, capillary electrophoresis, and capillary electrochromatography. Then some questions. So electroosmotic flow, when a high potential is applied across a capillary tube made of silica containing buffer solution, the positively charged ions migrate towards the negative electrode and carry solvent molecules in the same direction. As shown in the figure, the electric double layer is developed on the interface of silica and solution. The surface of the silicate glass capillary contains negatively charged functional groups, one formed due to the presence of silicon OH is there. So and they attract positively charged counter ions. This overall solvent movement is called electroosmotic flow. So during separation, the uncharged molecules move at the same velocity as the electroosmotic flow with very little separation. The positively charged ions move faster and negatively charged ions move slower. So they can be separated. The basic principle and operation. So in electrophoresis, so electrophoresis is a separation method based on differential rate of migration of charged species in a buffer solution across with a DC electric field is applied. An electrophoresis separation. An electrophoresis separation is achieved by injection of a small volume of sample into an aqueous buffer solution, which is contained on a porous support medium like paper or semi-solid gel or a narrow tube. So the simple schematic diagram of gel electrophoresis. The potential applied causes the species to migrate towards the one or the other electrode. The rate of migration of a given species depends upon the charge and also the size. Thus, the separation are based on the differences in charge to size ratio of the various species. Then what are the different forms of electrophoresis? Electrophoresis can be one-dimensional, meaning one plane of separation, or two-dimensional, meaning two planes of separation. One-dimensional electrophoresis is used for most routine protein and nucleic acid separations, whereas two-dimensional separation of proteins is used for fingerprinting, proteomic studies, etc. So different types of electrophoresis again can be categorized as slab electrophoresis and capillary electrophoresis. So what is slab electrophoresis? In slab electrophoresis, separations are carried out on a thin flat layer, that means slab of porous semi-solid gel containing an aqueous buffer. In general, slab has dimensions of a few centimeters on a side and is capable of separating more than one sample simultaneously. Samples can be very small as few microliter are introduced as bands or spots on the slab, and finally the separated species are detected by staining. Typical examples are DNA gel electrophoresis, SDS PEG, PEG gel electrophoresis, and two-dimensional SDS PEG gel electrophoresis, etc. So, what is DNA gel electrophoresis? In this technique, DNA samples are run through agarose gel with the help of a potential and differential mobility of different DNA samples is responsible for their separation. So these are the instrumentation. The following instruments and reagents are important for preparation and running of DNA gel electrophoresis and the gel casting trays. These are available in a variety of size and are composed of U-shaped UV transparent plastic. The open end of this tray are closed with tape while the gel is being cast in between A and B walls. The tape is removed before electrophoresis. Then combs. Okay. These are plastic combs around which molten agarose is poured to form sample well in the gel. Once the gel is cast, comb is easily removed and the wells are made in the gel slab. The size of the comb determines the volume of the well produced. So the comb means in a regular interval we have to make the well. Then electrophoresis buffer. The buffer which is used for DNA gel electrophoresis is usually tris acetate EDTA or tris borate EDTA. In the loading buffer, this buffer is important not only for loading of the sample in the wells of DNA gel but also to impart a color for the tracking of the sample. The components of the loading buffer are glycerol and dye. Glycerol helps to allow the sample to go into the sample wells, and the dyes help visual monitoring of the sample and the extent of electrophoresis has proceeds. Then ethidium bromide, which is used for detection of DNA inside the gel. Ethidium bromide is a fluorescent dye, giving fluorescence when intercalated inside the DNA and is used for staining nucleic acids. Ethidium bromide is carcinogenic and should be handled as a hazardous chemical, and one should wear gloves while handling it. Trans illuminator, it is the ultraviolet light source which is used to visualize the ethidium bromide stained DNA gel. So after GN electrophoresis is run, the whole slab is kept over trans illuminator. The spots of DNA in the agarose slab can easily be found by fluorescent intercalated lithium bromide as shown here. So here we have made the well and we have given the sample, so that we are giving the stains here. So B represents the glass gel slab before and after run respectively. The well M contains the standard marker to examine the mass of DNA. Then the SDS PAG gel electrophoresis. In this technique, protein samples are run through phase gel with the help of potential and differential mobility of different protein samples is responsible for their separation according to the mass of the protein. Polyacrylamide gels are formed from polymerization of two compounds, acrylamide and N,N-methylene bisacrylamide. Bisacrylamide is a cross-linking agent. Already we have discussed, I think, yesterday. The polymerization is initiated by addition of ammonium persulfate along with either dimethylamino-propionitrile (DMAP) or NNNN tetramethyl ethyl ethylene diamine tetramethyl ethylene diamine. Gels are cast in between two glass plates and combs are also used to prepare the wells. Like DNA gel casting, combs are removed after polymerization of the polymer. The gels are neutral, hydrophilic, three-dimensional networks of long hydrocarbons cross-linked by methylene group. Acrylamide and methylene bisacrylamide react to present in presence of ammonium persulfate to give the polymer. The instruments and reagents important for phase gel are gel casting apparatus, loading buffer and running buffer. Casting of gel not there or not. Yeah. Casting of gel is done in such a way that the whole slab will have two portions. One portion at the top, which is called stacking gel. You can see here. So the portion at the top is called stacking gel, and the rest part, the bottom one, is called the running gel. When the detergent SDS, sodium dodecyl sulfate, is added to the protein during the sample preparation, protein becomes negatively charged by either by their attachment to the SDS anion. When separated on a polyacrylamide gel, the procedure is abbreviated as SDS PAGE, that is for sodium dodecyl sulfate and polyacrylamide gel electrophoresis. These negatively charged proteins are allowed to run through the polyacrylamide gel towards the positive electrode. The technique is a powerful tool for the identification of the protein as well as determination of mass of a protein. A known marker protein mixture is used to compare and determine the mass of protein. So this one is the known marker protein. Then these are the samples. So this is one dimensional. Then if you go to the two dimensional SDS phase gel electrophoresis. Why again gel? This is gel polyacrylamide gel electrophoresis, PAGE. GE for gel electrophoresis. No need to write again gel electrophoresis. So SDS phase. Okay. So each amino acid has its own isoelectric point. As you know, that is the PI. That is the electric current which is used to stop electrophoresis is known as isoelectric point, which will form a Joule ion. So Joule ion means it will neither move towards the positive or negative electrode. So that pH at which the Joule ion will be formed is known as isoelectric point. So each amino acid has its own isoelectric point, that is Pi. Pi is the pH at which the mobility of amino acid is zero. That means Joule ion will be formed. the mobility of amino acid is 0. Hence, proteins which comprise amino acids do not have a do have a characteristics pi value, that means isoelectric point. In two-dimensional gel electrophoresis, proteins are separated according to their isoelectric point, and then they are allowed to mix with SDS. Denatured proteins are then allowed to enter a polyacrylamide gel. The passage of protein by potential ultimately separates the protein according to their masses. So it will be separated. So this is a two-dimensional gel electrophoresis. Then next is capillary electrophoresis. So capillary electrophoresis is a more sensitive technique, and with the similar principle as electrophoresis, it could detect samples as small as a few nanoliter. Besides being more sensitive, this separation technique has high speed and high resolution. Instead of staining of the samples as in slab electrophoresis, there is one quantitative detector at the end of the electroosmotic flow or at the end of the capillary. The capillary can also be filled with a gel which eliminates the electroosmotic flow. Thus, the separation is accomplished as in conventional gel electrophoresis, but the capillary allows higher resolution, greater sensitivity, and online detection. The buffer filled fused silica capillary, which is typically used here, is 10 to 100 micrometer in internal diameter and 40 to 100 centimeter long. It extends between two buffer reservoirs as shown in the figure, and also hold platinum electrodes. Sample introduction is performed at one end and the detection at the other. The polarity of the high voltage power supply can be made available, which can be reversed also to allow rapid separation of anions. So let us now study more about the sample introduction and detection in capillary electrophoresis. The sample introduction, the most common sample introduction methods are electrokinetic injection and pressure injection. With electrokinetic injection, one end of the capillary and its electrode are removed from their buffer compartment and placed in a small cup containing the sample. A potential is then applied for a period of time, causing the sample to enter the capillary by a combination of ionic migration and electroosmotic flow. The capillary end and the electrode are then placed back to the regular buffer solution for the duration of the separation. The injection technique discriminates by injecting larger amounts of more mobile ions relative to the slower moving ions. Then with pressure injection, the sample introduction end of the capillary is also placed momentarily into a small cup containing the sample, and a pressure difference is then used. In that case, you are using the voltage difference, potential difference. Here you are using the pressure difference. So pressure difference is used to drive the sample solution into the capillary. The pressure difference can come from applying a vacuum at the detector end, by pressurizing the sample, or by elevating the sample pressure in the sample end. So, pressure injection does not discriminate due to ion mobility, but cannot be used in gel-filled capillary. For both the injection procedure, the volume injected is controlled by duration of injection. Injections of 5 to 50 nanoliter are common, and the injection of volumes below 100 picoliter also has been reported. In the detection, there are several ways one can detect the sample after capillary electrophoresis. Examples are absorbance method, fluorescence method, mass spectroscopy, conductivity measurement, potentiometry, amperometry, radiometry, electrochemical detection, all these things. Then the capillary zone electrophoresis. Capillary zone electrophoresis, also known as free solution capillary electrophoresis, is the simplest form of capillary electrophoresis. It can be used to separate ionic species by their charge and frictional forces. And capillary gel electrophoresis is capillary zone electrophoresis. Then capillary gel electrophoresis, it is generally performed in a porous gel polymer matrix, the pores of which contain a buffer mixture in which the separation is carried out. In early slab electrophoresis studies, the primary purpose of the polymeric medium was to reduce the analyte dispersion by convection and diffusion and to provide a medium that could be conventionally handled for detection and scaling. Then the most common type of gel used in electrophoresis, that is capillary gel electrophoresis, is polyacrylamide polymer formed by polymerizing acrylamide in presence of cross-linking agent through infrared radiation. The pore size of polymer depends upon the ratio of monomer and cross-linking agent. Then capillary isotachophoresis. Isotachophoresis is a focusing technique based on the migration of the sample components between leading and terminating electrolytes. Solutes having mobilities intermediate to those of the leading and terminating electrolytes stack into sharp focus zones. Although it is used as a mode of separation, transient isotachophoresis has been used primarily as a sample concentration technique. Then capillary isoelectric focusing. This technique allows amphoteric molecules such as proteins to be separated by electrophoresis in a pH gradient generated between the cathode and anode. So, depending upon the pH, it will be separated. Then capillary electrochromatography. It is a hybrid separation method that couples high separation efficiency of CZE, that means capillary zone electrophoresis, and HPLC. So, capillary zone electrophoresis and HPLC. So, it is a combined method which is known as capillary electrochromatography and uses an electric field rather than hydraulic pressure to propel the mobile phase to a packed bed because there is minimal back pressure. It is possible to use small diameter packings and achieve very high efficiencies. So, electrochromatography, a hybrid capillary electrophoresis and SPLC, offers some of the best features of each of the two techniques. Two types of capillary electrochromatography have been developed since 1980s: packed column and micellar electrokinetic capillary. So, this is having very widespread application. So, this is all about the electrophoresis, and there are some questions and answers that you have to go through it. So, this ends your the subject, that is the separation methods. Okay. Thank you and best of your luck for the forthcoming exam.
Thank you, sir. Thank you, sir.
Oh, thank you.